I'd like to begin first by thanking Dr. Nihal Thomas for inviting me to chair this session. It is indeed an honor to be back in CMC for any scientific meeting. So the last time I came here was a couple of years ago when I gave a talk at the Medicine Annual Conference. So it is indeed my pleasure again to introduce another speaker from the same department where I finished my PhD in 2003, Dr. A.B. Abraham, who is currently a professor and heading the department, and also following the legacy of Dr. Maman Chandy. I'm glad to find that the legacy continues both in the clinical front as well as in the research. Dr. Ebi's research interest is in the inherited bleeding disorders and especially the hemophilias. And today's talk will be on lentiviral gene therapy for hemophilia A, an update on the clinical trial. Over to you.
Thank you, Dr. Everett, for the kind introduction. I thank Dr. Nihal and his colleagues in CSCR for the opportunity to present this topic. These are the disclosures, and this is the outline of my presentation. So, why gene therapy for hemophilia? It has got certain characteristics which actually make it attractive for gene therapy. It's a monogenic disorder and it results in deficiency of a single protein. And the complications of the deficiency of this protein, especially in those with severe, hemophilia can be classified as mild, moderate and severe depending on the level of the protein. The severe ones have less than 1% in the peripheral blood, in the plasma, and moderate between 1 and 5, and the mild between 5 and 40%. And these people tend to bleed spontaneously of varying degrees. The severe ones bleed almost a week, especially into their joints. There can be phenotypic variation in the severe group. And the lifelong replacement of the protein actually mitigate the problems of bleeding to some extent. That's called the prophylaxis. And the cost is very high, especially in developing countries. Accessing this drug is difficult. So there have been advances in terms of other ways of treating these patients, including non-factor therapies, where either a factor analog or factor mimetic, like bispecific antibodies, can act like factor VIII and therefore mitigate some of these issues. Also, there are rebalancing agents. As you know, the coagulation is balance between procoagulant and anticoagulant proteins, a fine balance in the body. And if in patients with deficiency in coagulation, procoagulant proteins, it can be rebalanced by knocking off the anticoagulant proteins, which actually lead to rebalancing and more of thrombin production. However, the cost of these agents, the administration issues like daily, or in children it's especially difficult to make them undergo injection almost on a daily basis. And the factor levels could be subnormal levels. It may not achieve the moderate or mild levels or even normal levels. And complications of replacement could include inhibitor formation, which is antibody against factor VIII or factor IX, which will render those agents ineffective. And therefore, it is a fit case for gene therapy. In gene therapy, there are multiple vectors which are available, the integrating vectors and the episomal vectors. The retroviruses and lentiviruses form the integrating group, whereas adeno-associated virus, HSV, and adenovirus form the episomal non-integrating type. In hemophilia, actually hemophilia space, lentivirus and adeno-associated virus have shown maximum promise and gone into the clinical trial phase. The immunogenic potential of these two groups are low. They have got tropism for dividing and non-dividing cells, which make it quite attractive. And the limitation for lentivirus group include potential oncogenicity, whereas those for adeno-associated virus, it has got very small packaging capacity compared to the lentivirus. And many people in the normal population, many of those have high pre-existing immunity, and also children cannot have AAV vector-based gene therapy because their livers are small, because AAV target has got tropism for liver, and therefore once a liver becomes big in an adult, the effect of this AAV gene therapy might wear off. Around 10 to 12 years and more is considered the cutoff for AAV-based gene therapy in children. The major advantage of lentivirus-based therapy is long-term gene expression in dividing cells. The AAV include non-inflammatory, it is non-inflammatory and non-pathogenic. Coming back again, what are the approaches for gene therapy in hemophilia? The LV-based gene therapy, actually you have to collect the progenitor cells, and then plasmids are packaged into producer cells, and these target cells are transfused with the LV particle, and these retroviruses are able to kind of go into the stem cells and transfuse them. So before infusing back into the patient, you have to give a myeloablative treatment to this patient so that the host, the residual, the stem cells are knocked off, otherwise the body will reject the transfused stem cells, and therefore it is an inpatient procedure. And these LV modified cells which produce factor VIII infused back into the patient, they home in onto the marrow and start producing the factor VIII. Whereas AAV-based gene therapy, the transgene is packed into an AAV vector. The transgene expresses factor VIII. These non-enveloped viruses can be infused in the outpatient setting also, because it's almost like any other intravenous injection. And they have tropism for liver. There are several serotypes. The ones with tropism for liver will home in onto the liver, and endogenous synthesis and secretion of factor VIII or factor IX by hepatocytes will happen, and it will continue to produce factor VIII. A new technology which has come up is about it's about CRISPR-Cas9 based gene editing. This is in vivo gene editing compared to an ex vivo gene editing, which is CRISPR-Cas9 technology is known for. Here, the advantage is that the CRISPR-Cas9 will open up the DNA area of interest, and there will be an associated AAV transgene along with that. All these are packed together in a lipid nanoparticle, and this lipid nanoparticle can be infused into the patient. Again, as an outpatient setting, it could be done, and this will home in onto the hepatocyte because of the albumin targeted double-strand break. So the albumin targets the hepatocytes, and double-strand break is made by the CRISPR-Cas9, and the AAV transgene will home in onto that area and start integrate and start producing the factor VIII. So these are the three ones which have almost gone into the clinical stage. So the summary and benefits and challenges of the three gene therapy approaches include the ex vivo LV-based gene therapy is capable of transfusing dividing and non-dividing cells, has a potential to treat pediatric patients, lower levels of antibodies against vector, and therefore it's effective, relatively large packaging capacity. The challenges include significant toxicity related risk, semi-random integrations and off-target effects could be there. Currently pediatric patients are ineligible for treatment for participation because of the concern about integration and long-term effect. Whereas AAV-based has advantages of easily engineered genome and capsid, considered to be a non-integrating vector. Long-term data is being accrued. It has got the maximum data available in hemophilia space, demonstrating efficacy and tolerability. Challenges include high seroprevalence against AAV viruses, and variable response and unknown durability. It has got potential off-target effects like integration, and pediatric patients are again ineligible because of the size of the liver becoming bigger as they become adults. CRISPR-Cas9 has got its benefits of transfusing dividing and non-dividing cells, has the potential to treat pediatric population and high degree of precision. The challenges include off-target effects, potential integration of AAV ITRs into double-strand break, and others include challenges of delivery systems, and pediatric patients are again ineligible to participate in trials unless there is long-term data in adults. So our journey in this space with lentiviral, actually CSCR had initiated both AAV as lentiviral gene-based approach. In fact, the CSCR had a collaboration with the University of Florida, Dr. Arun Srivastava, but what happened is it could not move forward because of certain reasons, and subsequently collaboration with Emory University had given this space, and here the heart of the thing is the lentiviral vector carrying a novel factor VIII transgene ET3. It is actually base source from porcine origin. The pigs have factor VIII expression, which produces much more factor VIII. Their gene construct actually expresses factor VIII much more than human factor VIII gene, and therefore this transgene, which when is incorporated into the human factor VIII gene, actually the resultant factor VIII product, the mRNA product, is able to express much more, and the factor VIII production is much more, and it has been found in the preclinical studies as well. So this is the heart of the concept. And preclinical studies in hemophilia A mouse had actually shown the enriched stem cells from these mice were transfused with clinical grade CD68ET3, and these mice were given radiation, lethal radiation, and then subsequently they were monitored for factor VIII ET3 activity, engraftment, and inhibitor formation. These studies actually demonstrated the goal of 5% factor VIII level. The 5% is the moderate level which I mentioned earlier. That's a target even in medical therapy. That's a target which we use because the moderate hemophilias actually bleed much less compared to severe hemophilia, because even a slight rise in factor VIII level from less than 1% to up to 1 to 5% makes a huge difference in the spontaneous bleeds. And these could be achieved in these mice, and with a kind of predictable vector copy number, which could be translated into meaningful factor VIII levels. And subsequently it was actually transduced, HSC transplant was done in immunodeficiency mice. Here again, GCSF mobilization, HSCs were mobilized, and these HSCs were transfused with vector construct, and therefore after that these mice who were immunodeficient 6 weeks old, hemophilia A or recipient NSC mice were conditioned with busulfan, and then these transfused cells were infused, and subsequently ET3 expression was monitored for, and it was again showed the same effect of good expression of factor VIII. So while with this promising approach we moved forward to the next level. While the lab work was going on, we had actually had extensive tour of Tamil Nadu. This was important because the hemophilia is a disorder which is benign, which is not immediately life-threatening. There are medical therapy available. Will patients be ready for groundbreaking treatment like this, which is first of its kind, which is not done anywhere in the world, at least in hemophilia with lentivirus therapy. So we had to really raise awareness in the patients, and we interacted closely with patient advocacy groups. We had conducted many camps, and we had education and community participation in a very active way. The team traveled all over Tamil Nadu. Actually, it went on for almost 1 to 2 years. Meanwhile, the preparation for the entire clinical program was made by the clinical team in hematology. The regulatory approval and for GMP and clinical were another bottleneck. I won't say bottleneck, it's the first time in the country, and therefore we had to, actually the regulators were not sure of what exactly they had to put in place, the rules, the regulations, and it took time, and that's but natural. It took almost 3 to 4 years for the regulators to kind of put in place the rules. The CDSCO finally granted the permission. So the clinical study protocol included part 1, the stem cell collection, and part 2, the conditioning. So the stem cell mobilization was done with GCSF and plerixafor, and then the apheresis was done. If there was actually enough cell CD34 selection, and transduction was done, and then cells were cryopreserved after quality checking. And the part 2 included the patient conditioning. We had conditioning which we had devised for our allogenic stem cell transplant, so based on fludarabine and triosulfan, and we infused the the PBSC cells, which is collected from the patient themselves, and then we monitored them. In fact, we did this, the patients were admitted in the allogenic stem cell transplant unit, which is HEPA filtered, just like any other allogenic transplant. We were very careful to make sure that, because this is a first in human trial, we wanted to make sure that nothing went wrong. In the lab, we had this Clinimax machine, which helped us in mobilization and apheresis, and labeling, washing, enrichment, and collection were done. And the clinical part included, the primary endpoints included the safety endpoints, 100-day survival, and immune response to ET3. The secondary endpoint included factor VIII activity, as well as bleeding episodes, and anybody developed inhibitors, time to neutrophil recovery, time to platelet recovery, because these are all myeloablative conditioning, which we monitor routinely in a stem cell transplant. We also monitored for vector copy number and vector integration profile. The protocol we used was GCS of 5 mg/kg per day, twice daily for 4 days, with the plerixafor the day before the apheresis. After collecting the CD34 cells, it was transduced, and then it was frozen, and then the patient was admitted and given the stem cell transplant condition, which included triosulfan for 3 days at 14 gram per meter square, fludarabine for 4 days at 30 gram per meter square, and then reinfusion of the transfused HSC on day 0, and waiting for the engraftment. We had actually 8 patients. Midway through, in the group 1, we realized that the P2 developed inhibitor. I'll come to that story later. So we could not enroll him. Just after conditioning, we had to abandon that. So P1 and P3 got the regular routine autologous HSC transfused cells without transduction enhancer. At that time, we realized that the factor VIII recovery in these patients were not, it's in the moderate range, and just in the moderate range. And therefore, Dr. Gurban Singh from CSCR had a brilliant idea. He actually devised a particular protocol which made the vector enhancement, vector transduction much more efficacious. And we subsequently found that in the preclinical stage it was very good, and therefore we brought in towards for the clinical trial as well. So for P4 to P8, we found that this alternate method actually transduction enhancer had increased the vector copy number, and the factor VIII levels were also much higher. So this work was published last year. Dr. Alok Srivastava had taken the lead in conducting the study and publishing this paper. So actually, the factor VIII levels in plasma at day 60 and up to last follow-up till September this year. This is a table which shows that the average age varied between 20 to 40 years. The factor VIII levels on day 60, you can see here. And the median range in the first 2 patients were lower, and subsequently with the enhancer we had much higher levels. This is a follow-up of these patients almost up to 3 years now, and the vector copy number is here. You can see that the vector copy number also increased in the latter group, and that corresponded with much higher levels of factor VIII. So this graph actually shows P1, P3. So P2 had to be withdrawn. P4 and P5 did not participate because of hypertension and patients' concerns about infertility. P2 had to be withdrawn after the 1 day of conditioning because of inhibitor development. So the rest of the patients, you can see that as the years, the months went by, the factor levels were steady, maintained steady. You can see that they are almost in the normal range. You can see in the normal 50%, at least 2 or 3 of them, and the rest in the mild or moderate range. The P2 is an interesting story. This patient, after admission and after being given factor VIII, developed inhibitors because on the day we could find that we gave 100% correction, but our recovery was only around 10%, which make a hematologist have palpitation. So subsequently we withdrew him from the study, and then we gave him immune tolerance induction, where we give high dose of factor VIII and the inhibitor becomes negative. And subsequently, after 1.5 to 2 years, we took him for gene therapy again, And subsequently, we found that almost after 1.5 years of follow-up, his levels are really good in the mild range, more than 20%. So this forms the first successful expression of measurable and sustained factor VIII activity in plasma after gene therapy for hemophilia A in the history of, with a history of inhibitors, and which are resolved with immune tolerance induction. So this is the first report in the literature in such a patient. And you can see that this is actually going into the mild range. As if to just to wait for the cell and gene therapy symposium to happen, this particular patient with inhibitor, who's the first patient to have this in the literature, he fell down on 4th and developed a fracture of both tibia and fibula of the right side. This happened just last week. So he is a severe hemophilia with low titer inhibitor. He was actually underwent gene therapy on 28/6/2024, 15 months post gene therapy. He's being followed up, and he fell down and sustained this injury. And we found that at presentation his level was 22%, chromogenic 27%. Baseline factor VIII level when he came to ED was around, emergency department was around 37%. So he underwent open reduction and internal fixation called Iliazarov fixator application on 7th. And these are his factor levels. Those who are interested in hematology clinical space will be able to understand this better. We use standard half-life factor VIII in these patients. We have long-acting factor VIII which we could use in these patients, which means once a day administration. Standard half-life we have to administer twice daily. Whereas for this patient, the baseline was 37, and we gave 30 international units, that is around 60% correction. And it really went up. And subsequently, we are using only once daily support and only around 30% correction. Whereas it will be double the correction which we usually give in severe patients, and also we have to administer standard half-life factor twice daily. So in this patient, half the dose of standard half-life and only once daily. So that's really impressive in this patient. Coming to the conclusions, this is the first in human lentiviral gene therapy for patients with severe hemophilia. It has shown efficacy and safety in the small number of patients studied. Even though promising long-term efficacy and safety need to be assessed, cost is a barrier for these patients in our country unless domestic manufacturing of reagents is scaled up. Stem cell-directed gene therapy is attractive in children, but the role of conditioning in these patients, because it's chemotherapy, Dr. Biju would have mentioned in the previous presentation about infertility and other long-term effects, whether their oncogenic potential, and therefore how you reconcile these in benign disorders is still a question. Alternate medical therapy in hemophilia patients have expanded exponentially, which I mentioned earlier, and therefore what will be the role of gene therapy, that also remains to be seen. So a lot of people to thank for the CSCR for actually doing this preclinical study and taking this forward. The collaboration with Emory University has made this a reality. INSTEM Bangalore for supporting through various means. The Department of Biotechnology, Government of India. The hematology department, the colleagues, both clinical and the paraclinical and lab side. Other staff, Dr. Alok, who had initially initiated this trial, and the clinical pathology and coagulation lab colleagues who have done the coagulation assays and other support. Thank you very much for your attention.
The floor is, thank you so much for the talk. The floor is open for questions.
Very impressive work and very good presentation, sir. My question is about the follow-up of these patients. Is there any plan of looking at the integration site characterization in the expanded population?
We had done it once last year. That did not show any concern. We'll continue to do that.
Thank you for showing us this landmark study. I mean, my thought is the moment I saw that fracture, that gave me some thoughts about when was the therapy for that specific patient who had the fall in the fracture? When did you actually do the myeloablative therapy and the lentiviral therapy?
It's
How many years? What's the gap?
So it's 1 year, 4 months.
That's pretty rapid to
Yes.
get an osteoporotic
Yes.
a fragility fracture like that in just a year and 4 months. And the age of the patient was
22.
22. So I'm just wondering, is there more to it with this integrative form of treatment, the lentivirus plus the myeloablative therapy, that has induced a fragility fracture so early? Very, very striking. 1 year is a bit too far. I don't expect even gonadetoxicity or anything to kind of induce osteoporosis so fast. Is there something else going on in the bone? Can a lentivirus have some primary effect on the bone?
Generally, most of the hemophilic patients have osteoporosis due to various reasons.
Sure.
One is that recurrent bleeds make them relatively inactive. Second is they themselves are scared of sustaining trauma, and therefore they are a bit careful about their physical activities are generally restricted.
Sure.
And also there is some evidence to show that factor VIII has actions beyond just coagulation. It probably acts on the bone, cartilage, and that is still an area to be explored. In fact, we are actually doing studies on that. We may be able to, we are trying to see if there is, what is the role of factor VIII in this space.
Yeah,
So
I'm
it's
just wondering whether you can actually do some special stains on the bone and
we could do that. We could do that. We could do that.
separate look
Yeah,
at that,
yeah.
and maybe even take a small piece of fresh bone and see
Yeah.
whether we can look at that in the
Yeah,
patient.
sure, yeah.
Thank you.
Thanks for the suggestion.
Thank you for
Hello,
the impressive talk
one
once
question
again.
here.
We have 2 questions online, so maybe you can have, you can catch up with them during the tea break. The first one is, okay, ET3 is a transgenic synthetic gene you have incorporated in lentiviral vector. How did you validate the function of ET3, and what are the modifications you have performed in factor VIII gene to derive ET3? That's the first question.
So this was done by the Emory group. So I think Dr. Gurubin can answer this. He is there in the audience.
Yeah, so just to answer that part, we have this factor VIII, which is having a sequence for secretion from the porcine sequence, and then we have the human sequence. So that's how it's a hybrid molecule which has high secretory efficiency as compared to human factor VIII. So that's how it was derived. It's a fusion protein and it was put into the lentiviral vector.
I hope that addressed that question. So next one is oncogenicity risk in patients treated with lentiviral gene therapy. What is a recommended follow-up period?
We are planning to follow for at least 15 years, maybe even more. As per protocol, minimum 15 years.
So do we need fludarabine for an autologous transplant? I mean, whether we can do away in the next phase just with myeloablation?
It's a possibility, but before that we should probably be doing some preclinical studies because this is a standard practice we have been using, fludarabine triosulfan. Therefore, we just followed it.
Dr. AB, I know this was done under a clinical trial funded. So if you were to do it for patient, what is the cost of the gene therapy protocol?
In fact, the cost of medical care is not that much. A standard, if we don't necessarily have to admit these patients in HEPA-filtered room. If you admit them in HEPA-filtered room and do it, it will cost something like 10-12 lakh rupees, maybe slightly more. They don't develop much complications like other patients, and therefore we don't need high-end antibiotics. In fact, it could even be brought down to around 8 lakhs, medical part of it. But the lentiviral vector and all those things, that is the main bottleneck. That's because it is manufactured, it's not being manufactured in the country. There is facility, actually one company in Faridabad will do that, but even for them, the reagents have to be imported. So many of these reagents are manufactured in the West. That is the main bottleneck. So if we are looking at minimizing the cost to the maximum possible, and there are several ways of doing that.
Okay, so one question mandatory from my side because I'm moderating his talk. I'm not sure whether I missed this one. What about factor VIII dose tapering? Because how long did you have that after the gene infusion? Like Normally, we don't abruptly stop the factor VIII replacement therapy because we don't know whether gene therapy would work. So until it kicks in, we would keep it for some time. So I probably missed it on the talk, but then
Yes.
yeah, this dose tapering, how long did it take before you actually stopped it eventually when you saw signs of gene therapy kicking in?
So these patients are potentially can have mucositis, which means denuding of the oral mucosa, and could potentially bleed even from the gut. And therefore, we continued for at least 10 days post-infusion. And neutrophil and platelet engraftment happens in this patient. That's a surrogate marker for us to say that probably the stem cells have engrafted, and therefore have started producing the factor VIII. And therefore, by around 10-14 days, we'll support. After that, we'll switch to prophylaxis, which means once or twice a week. And then we'll probably stop by around 3 weeks. And we have found that factor VIII production, the exogenous production, the transfused cells have started producing by around 3 to 4 weeks. As early as 2nd to 3rd week we have found.
Thank you so much for your talk.